This briefing decrypts the biological blueprint of human behavior, starting with the physical mechanisms that dictate what we mistake for intent. The rabies virus provides a clear window into this architecture. It operates as a mechanistic software glitch that hijacks the host's primary operating system. By navigating specifically to the limbic system, the virus triggers a controlled cascade of aggressive neurochemicals. It effectively overwrites standard behavioral algorithms with a singular, high-intensity output. Even the characteristic fear of water is a calculated structural strategy. By inducing throat spasms that prevent swallowing, the virus remains concentrated in the salivary glands, ensuring its own mechanical transmission via a bite. We observe similar systemic overrides in clinical vampirism, or Renfield syndrome. In these cases, a malfunction in the brain's dopamine feedback loop creates an obsessive compulsion to consume blood. The behavior is driven by a broken reward circuit rather than a psychological or moral deviation. Throughout history, these chemical failures were often mythologized. Porphyria is perhaps the most frequent historical source for the vampire archetype. Porphyria is a breakdown in hemoglobin synthesis that causes a toxic buildup of porphyrins in the blood. This results in extreme photosensitivity and a physical receding of the gums that gives the appearance of prominent fangs. These cases demonstrate that human behavior is susceptible to chemical override because the will itself is an expression of these thermodynamic systems. Project Genesis posits that the transition from inorganic matter to the human mind is a continuous physical reaction propelled by the laws of thermodynamic equilibrium. We define life as a self-sustaining chemical system capable of Darwinian evolution, relying on the persistent storage and replication of data. This timeline tracks the 4 billion-year progression from the formation of Earth and the appearance of the first microfossils to the modern era. We are tracking the mechanistic steps that lead from a primordial soup of gases to the emergence of a processing network composed of 86 billion neurons. Under this framework, humanity represents the most complex iteration of a 4 billion-year-old, entirely unguided chemical chain reaction. Early Earth's atmosphere was a volatile mixture of simple, carbon-rich gases. High-frequency electrical discharges, like those seen in this Miller-Urey simulation, physically shattered these gas molecules, forcing their constituent atoms to recombine. This continuous thermal and electrical bombardment abiotically forged the first amino acids and nucleotides, the primary building blocks of life. However, these molecules cannot spontaneously assemble into complex, ordered chains while floating randomly in a vast ocean. The process required a structural scaffold to coordinate the assembly. This was provided by crystalline mineral surfaces. Mineral faces act as organizing templates through electromagnetic attraction. Here, oxygen atoms in amino acids lock directly onto the calcium atoms of a calcite surface. This rigid crystal lattice forces molecules into highly organized two-dimensional structures, providing the necessary proximity for polymerization. The foundational organization of life's building blocks was dictated by the mathematical geometry of the rocks and minerals that preceded them. To maintain the integrity of these early polymers, the system utilized lipid molecules as a primary hardware housing. Each lipid molecule possesses a distinct physical polarity, an electrically charged head and an uncharged hydrophobic tail. In a watery environment, the charged heads are pulled toward the fluid by electrostatic attraction. Simultaneously, hydrophobic tails are forcefully repelled. To achieve minimal energy, these lipids autonomously assemble into a perfect spherical barrier, tails pointing inward, away from water. This geometric structure, known as a vesicle, establishes the first physical boundary between a system and its environment. These early membranes faced an immediate environmental threat: charged magnesium ions in the ocean that physically shattered fragile lipid structures. The solution lay in the amino acids already present within the primordial soup. As amino acids bind to the surface of the vesicle, they act as a physical buffer, stabilizing the membrane against the disruptive effects of the magnesium. Cellular boundaries emerge as a byproduct of hydrophobic physics, providing structural stabilization long before biological protection was an evolutionary factor. Early RNA served as the system's cloning mechanism, relying on the electrostatic attraction between base pairs, adenine to uracil and cytosine to guanine. This architecture contained a critical vulnerability centered on the base cytosine. When cytosine collides with water, it undergoes deamination. This chemical reaction pathway shows cytosine physically transforming into uracil. This collision corrupts the sequence data, changing every C into a U, and effectively destroying the information needed for replication. To resolve this, the system transitioned to the more stable architecture of DNA. This involved a subtle physical modification, the attachment of a methyl group, a specific chemical fragment, onto the uracil structure. This modified, heavier structure is known as thymine. By utilizing thymine instead of uracil, the system's chemical proofreaders can instantly identify any uracil base as a structural error and execute an automated repair. DNA serves as the system's mechanical resolution to data corruption, providing the high-fidelity stability required for complex life. At the cellular level, the biological concepts of hunger or hunting disappear. Cellular intake is driven entirely by blind structural binding events on the cell's exterior. Kinetic energy from water currents, known as Brownian motion, pushes external molecules arbitrarily against the cell surface. Only molecules with an exact three-dimensional structural match and an opposing electrical charge can bind to the protein receptors. This geometric complementarity dictates survival. Cells that possessed the matching receptors absorbed material and persisted. Those that did not simply dissolved back into the environment. Sustenance is the mathematical probability of electrostatic binding across a surface. This mechanical logic reaches its peak in the neuron, a specialized binary electrochemical relay switch. A single isolated neuron is devoid of thought or awareness. It is a discrete physical component. Its function is triggered by an electrical spike that opens physical gates on its membrane. This opening causes a flush of calcium ions to surge across the synaptic gap, physically striking the receptors on the adjacent cell. The impact of these ions forces the adjacent cell's receptors to open mechanically. This forces the second cell to discharge its own pulse, creating a chemical chain reaction. This is the biological domino effect. When we scale this binary mechanism, we see the architecture of the human brain. This exact binary trigger repeats across a network of 86 billion interconnected neural nodes. The sheer speed and density of these electrical transfers creates a massive processing volume. Biological computation is the aggregate result of these billions of cascading triggers interacting in real time. Consciousness represents the aggregate output of 86 billion structured triggers. At this scale, the cascading physics of the network becomes the experience of the cell.